All right, here is your rubric for WA3. Here's how you need to look at that. Realize that in WA3 you're going to have headings, so you'll have an introduction, and you'll need to put a heading at the top of a page. You'll need materials and methods, results. You'll need questions, legends, and they need to be in the for the figures and legends. Now up here, introduction, you've already WA1. The one thing we need to talk a little bit more about is our model organisms and what they are, and whether or not Drosophila makes a good model organism. Sometimes it does. If you're studying, say, human medicine, and you're trying to understand how the human body works, you would, if it translates to Drosophila, but Drosophila is an insect, so it has to be a very simple mechanism that you're studying. Something complex that's only true in humans isn't going to be, you can't study that in Drosophila. Maybe you need a chimp, or maybe you can get away with a mouse. But insects have very tiny brains, just a small ganglia of a few ganglia of neurons, so it's not like they have a complex brain. But maybe that's something that you want to simplify that if you're studying that. But other things, a lot of things like pigmentation and things, easy to study in Drosophila versus humans. And as a general model, it makes a good model. And you have to know that a good model organism is, of course, cheap, and they don't take up a lot of space, things like that. Those are the things you need to talk about. All right. You, of course, need to include how the mutation was discovered and the other 4 related genes. That's more or less what you did in WA1. And that gives you a page or 2. So you really only need to add like another page. You do need to elaborate more on the biological processes involved in your mutant's function here. But in general, you don't have a lot to do in the introduction, so it's only another page or so, and it doesn't count much over here. Materials and methods also doesn't count much, mainly because in the way we're doing it, you don't have as much to do here as you did in previous semesters. We only use Fly Labs, so you don't have to talk about all the equipment as much, but you do have to discuss how to do the statistics. Now, you don't talk about your results here, you just talk about like how you do a chi-square, how you do crosses, that sort of thing. So you do need to put that in. And here in results, now you begin to talk about what you found and your hypothesis, and you kind of you don't put it together yet, that's for the discussion, but you do list your results. So you might give tables and stuff here. You can put the tables at the end or in here. Pictures go at the end for sure in figures, but here if you wanted to put tables, I'm not going to count off if you put your tables here, if they have to do with chi-square. But you need to talk about how crosses are done and what you found. Is it sex-linked, that sort of thing. So that's what's going to come up here, and all your results from at least the work you've done. You haven't done the bioinformatics yet, but you will. All right, now in the last part is where you discuss it. So you take all your results here, kind of put it all together. You might have several different results from your various crosses, your various experiments. And you have to put it all together and give an overall conclusion as to what you think is going on and what the data means. So you've got your overall discussion about how everything works. And that's going to be 3 to 4 pages. It'll get a little longer with WA5. References, make sure you number them. Start at the top of a page for references. So it needs to be a brand new page, references at the top, number them 1 through whatever, 6 to 10 at this point, and left and right justify them. Make sure you remember that. A few people did the left but didn't do the right. And then down here is where you put your figures and legends. This is where you might be doing most of your work for this, and it reflects in the fact that it's worth 25 points. Recognize the majority of the points you're going to be doing in the results part, where you list all your things. So you kind of take WA2 and kind of get going with the results and expand on it on on the materials and methods and the results from WA2. Uh, Down here with so you'll have lots of pictures to draw. I would suggest having more than one figure from WA1. If you did one and you did really well on it, fine, but you might want to get another picture of your fly and put more than one in. But here, these are all your tables, your crosses, your pictures of your chromosome mapping, that sort of thing. And that should do it. Basically, you're taking WA1 and WA2, combining them, and putting them in a form where you can get ready for WA5. Most of your work here should be in expanding the introduction. Also, organizing your results from WA1, talking about, say, how chi-square works here and how FlyLab works, how the crosses were conducted, but that's still FlyLab. Here are where you discuss your results. So you can probably put a lot of tables in here with your results in it. And I do allow you to put tables in your results if you want. You can put them at the end and refer to them, or you can put them in here and talk about them if you would like. And then finally, your discussion is 3 to 4 pages. Most of your points are here and down here at the in the figures. All right, that should do it.